Hello, welcome back. This is Dr. Balding. The last section that we talked about on microbial genetics was we were trying to understand some of the basic genetic processes that occur that were originally identified in prokaryotic cells. So our focus in our last lecture video series and class was DNA replication. Tonight we're going to talk about these other processes that were originally part of the central dogma of genetics, right? How DNA goes to RNA, how RNA is made into protein. But we now know that there are enzymes out there that sort of go against the central dogma that RNA can be made into DNA through some processes in viruses. We have RNA that can also be transcribed into other RNA molecules in viruses, but we'll come back to those processes when we talk about the virology section. But right now, we're going to start with this process of transcription, where we make an RNA molecule, right, from DNA. Okay, any questions that you guys have about DNA replication from the videos that you watched? No? Okay, if you do, just let me know. Okay, so we're going to talk about the process of transcription. Transcription, right, is going to occur using an enzyme that we call RNA polymerase, right? It's functionally similar to DNA polymerase in that it needs it makes a complementary copy of nucleic acid from a template molecule. There's some differences though. Instead of copying long DNA sequences, we're copying short segments of DNA or gene regions, right? The RNA polymerase is going to be able to recognize RNA nucleotides, that is ribonucleotides, rather than deoxyribonucleotides. So you see the incorporation of uracil, okay, you guys know that, rather than thymine. RNA polymerase is also going to do things a little bit differently. One thing is that it's going to go much slower than DNA polymerase. DNA polymerase processes nucleotides at about 1,000 nucleotides per second. So it's moving really quickly. And you may say, why is it moving so quickly? Well, it moves so quickly because it's copying whole chromosomes, right? And so it has to do it rather fast, right? When we are copying DNA into these short RNA sequences, right, it doesn't have to occur as quickly. So RNA polymerase works at about 50 nucleotides per second, right? So not quite as fast. Also, RNA polymerase does some things the same. It works in the 5' to 3' direction, adding new nucleotides to the 3' hydroxyl end of the DNA. All right, I'm sorry, to the new growing RNA transcript. Okay, so it works in the same direction as RNA polymerase. Another way in which it is different to the 3' hydroxyl end of a growing RNA. Another way in which it's different is that RNA polymerase is going to have no proofreading ability. If you guys remember when we talked about DNA polymerase, we said that it has an exonuclease activity, right? So it can actually, when there's a mistake made, back up and fix that mistake and then move on. But with RNA polymerase, you don't get proofreading. It doesn't have an exonuclease. It can't chew back. And so we end up making some RNA copies that may be wrong. Is that a problem? What do you guys think? It actually doesn't end up being much of a problem, because with that, if you make a mistake in one RNA, that protein that's made, it may be non-functional, right? It may have actually better function depending on what mutation was made. Okay, that's unlikely, but it could. But there are presumably plenty of copies that will be correct, and whenever you transcribe a gene, it's not like making another copy that's going to go on to a daughter cell. Like that's the only copy of that gene maybe that daughter cell gets, right? In this case, when you transcribe a gene into RNA, you get thousands of copies usually of that particular RNA, and some of them are going to be correct. And in fact, a whole abundance of them will be correct, and so you'll make the protein that you need. So it's all right if you make a few mistakes. And so RNA polymerase doesn't have what we call the fidelity, right? The ability to incorporate the correct nucleotides into a growing RNA. Another thing that RNA polymerase has that DNA polymerase doesn't is helicase activity. Okay, what's a helicase? Yeah, so it unwinds the DNA. So DNA polymerase requires a separate helicase to come in and unwind the DNA when the DNA is going to be replicated. But RNA polymerase can actually do that job itself. So it functions to unwind the DNA when it needs to. All right? So those are some of the ways in which DNA polymerases and RNA polymerases are similar and different. Okay, so let's look at what RNA polymerase does. So it's going to recognize a particular gene sequence and transcribe it. How does it know where to start? You guys know? A promoter region, right? There's a specific DNA sequence that RNA polymerase can recognize that we call the promoter, okay? RNA polymerase binds to something that we call the Pribnow box. In bacteria, and it's also called the -10 region. Why would it be called the -10? It's actually 10 nucleotides upstream of the start of transcription. That's why it's called the -10. Okay, so there's usually a specific sequence there that's called the Pribnow box that RNA polymerase can bind to. All right, it's found in the promoter. Now, so that's how the RNA polymerase binds to the gene sequence that it's going to transcribe. How does it know which genes to transcribe? Well, that's actually controlled by transcription factors. And the first transcription factors were actually found in prokaryotes. They're called sigma factors, okay? And so you guys remember back early on, I showed you an endospore. And when I showed you the endospore, I said there's about 300 genes required to make this endospore, all right? And then I said, when we make an endospore, it has to be turned on by something, right? Nutrient deprivation or desiccation, drying out, right? Something has to happen in order for that endospore to get turned on, right? Or the genes to be turned on. And so there's a cascade of sigma factors that are actually produced to turn on the genes required to make that endospore. And so that's how prokaryotes, and eukaryotes too, actually regulate the, simultaneous expression of genes needed in a particular process. They express these global regulators that turn on genes. Now, the one that turns on the housekeeping genes are called sigma 70s. Okay? So the sigma factor, usually if it's a constitutive gene, meaning it's going to be on all the time in the cell, we use something called sigma 70. Other sigma factors are required for other processes, right? So there's one involved in a heat shock process. If you heat shock prokaryotic cells, they'll express that particular sigma factor. If you need to produce an endospore, there's a whole cascade of them that are produced. There's one for iron limitation. There's one for nitrogen limitations. So there's a whole bunch of these that are expressed only under certain conditions, right? And so that's how these genes get turned on. So now the sigma factor, what does it do? It binds to a particular region upstream of that gene that's called the minus 35 region. So the minus 35 is upstream of the minus 10, and it helps direct RNA polymerase to that promoter sequence. The RNA polymerase can then interact with the minus 10 region, and if everything's good, right, there's nothing blocking that promoter like an operator, a bound operator sequence, right? Then RNA polymerase can proceed, and like I said, it's got helicase activity, so it can unwind the DNA and start moving along the length of it. Okay, this is called the initiation of transcription. Okay, so that's the first step, the initiation. It involves the sigma factor binding the RNA polymerase, it helping the RNA polymerase find the particular gene or genes that need to be turned on, and then the opening of the DNA, okay, with its helicase activity. The next step is elongation of the chain. So at the start site of transcription, right, RNA polymerase will start adding on nucleotides that are complementary to the DNA sequence it's copying, okay? And so you see it moving in this direction with new RNA ribonucleotides being added by dehydration synthesis. You guys remember, a hydroxyl's taken off one nucleotide, a hydrogen off the other, and they're joined together by a covalent bond in a phosphodiester bond, right? And water's lost. Okay, so you end up producing a water in this process. And you move along the length. And so then what also happens is the sigma factor falls off the RNA polymerase. Once it's used for initiation, that global regulator doesn't need to be there anymore. It can go bind another RNA polymerase and help it find an appropriate gene to bind to. Okay, so they get reused over and over again. In eukaryotes, it's a little more complex. If you guys have taken molecular biology or will take molecular, you'll talk about the more complex processes that occur in eukaryotic systems and a cascade of transcription factors. And oftentimes the concentration of transcription factors that will allow certain genes to be transcribed at given points in time or in given parts of the cell. So it gets a little, once you move into the eukaryote, it gets a little more complicated than just is the sigma factor present or not, right? Eukaryotic cells don't have sigma factors, they have transcription factors, but generally there's more than one regulating a particular process, okay? So it gets a little more complicated. Now, this is the elongation. That's the middle step. The last part is termination. How do we know when this transcription should stop? Why doesn't RNA polymerase just keep on going? Well, it reaches what we call a terminator sequence. Okay? And there's really two types of terminators that have been discovered in prokaryotes. There's what we call self-termination or independent termination, right? And it is sequence dependent. So if you guys remember when we were talking about nucleotides, right, in the beginning, and I mentioned in your video that there were 3 hydrogen bonds between cytosine and guanine, right? And there's how many between adenine and thymine? You guys remember? Is there 3? There's 2, okay? There's 2. So there's 3 between GC pairs, and there's 2 between AT pairs. So which ones are more stable? GC. So if you've got GC pairs, they're held together more tightly. So what happens is we have RNA polymerase is coming across a sequence that's very, very GC rich. Okay, and that's what the terminators look like. They're GC rich. And RNA polymerase slows down. Okay, that slowing down actually allows the RNA, the new RNA that's made, to form a stem loop structure. Okay, which can destabilize the RNA polymerase. And then all of a sudden, you see a whole bunch of T's and A's. Right? You see this sequence is full of T's and A's there in the terminator? And what happens is RNA polymerase is destabilized by this stem loop, this bulky stem loop structure, and then it speeds up. And it's believed that that mechanically kind of kicks off the RNA polymerase, right? And allows the RNA transcript to be removed, right, from the complementary DNA. So the DNA rebinds or rehybridizes the 2 strands, and the RNA polymerase comes off. Okay, so that's a self-termination event. Now we also have what we call a protein-dependent termination or enzyme-dependent. So when scientists actually started looking at genes in prokaryotes, and they were looking at a whole bunch of them, they realized that not all prokaryotic genes have the right terminator sequence to form this stem loop, right? And then they found a sequence that was common among other gene sequences. And so then after looking at it for a while, they found an enzyme that could actually wrap around the new RNA sequence. It's called Rho. And kind of chase the RNA polymerase along. And when RNA polymerase gets to this terminator sequence that's over here somewhere, right, it slows down and it interacts with this region called RUT. Okay? So that DNA sequence actually slows RNA polymerase down. Rho has the ability to come up and catch up with RNA polymerase, and it will destabilize it at that specific sequence. Okay? It's believed that it's a structural type of interaction, right, that causes that destabilization there at the RUT site. Okay? So that's an enzyme dependent, where the enzyme sort of row kind of follows along the RNA transcript and kicks RNA polymerase off. And then, like I said, we have self-termination where it's sequence dependent. There is no enzyme there that does it, right? Rho actually uses ATP to do this, so it's sort of energetically kind of unfavorable. So we find a lot more of these self-termination, which doesn't require energy, right, types of sequences in the cell than we do Rho dependent. All right, so if we look at transcription doesn't just occur, on one transcript, a single RNA at one time. A lot of students would think this is a snapshot or basically a series of events shown in this picture, right? So a whole bunch of, or one polymerase at different points in time, but that's not actually what this picture is showing. Each one of these are unique RNA polymerases. So here's one RNA polymerase, 2, 3, 4, 5, 6. The sixth one started transcription, okay, proceeded along, and then 5 here came and jumped on that promoter and started transcription right after it. So this is like one snapshot in time. So you can have many, many RNA polymerases transcribing a single gene at a single point in time, okay? And what's interesting is that, remember, in the prokaryote, there's no separation, no compartmentalization of transcription and translation. So as soon as this new RNA down here has a leader sequence and what we call a Shine-Dalgarno sequence, which is a ribosomal binding site, ribosomes can actually hop onto this transcript and start translating it into protein. And you can have not just one ribosome, but multiple ribosomes. So what you end up with in the nucleoid of a prokaryote is sort of this lamp brush structure of transcription and translation occurring all at one time in the same cell soup. And I know in the beginning I made a big deal out of compartmentalization in true nucleus. And this is one of the reasons, right, is that you don't end up with the separation of these two processes. So regulation occurs very differently in prokaryotes than it does in eukaryotes. A lot of the structure and a lot of the enzymatic processes of, putting the nucleotides together work the same. But a lot of the regulatory processes that control these activities can't occur in the same way because they're not separated into compartments, right? This is all occurring in the cell soup in a prokaryote, whereas in a eukaryote, we get compartmentalization. All right, so let's talk about compartmentalization. Compartmentalization allows for certain things to happen in a eukaryote that don't, like I said, in a prokaryote, and one of them is mRNA processing. Okay, so back long ago, you probably heard of introns and exons. Have anyone heard those words before? My molecular students better say, yes, Dr. Balding, I've heard of those things before because you talked about them, right? Yeah, so nod your heads. Yes, you did. Okay, some of you guys, I don't know if you have or not, but what happens in eukaryotes is when you look at a gene in a eukaryote, there's certain parts of it that are going to encode for messenger RNA that will encode for protein, right? And there's certain regions that are not going to be translated, okay? So they're labeled in this diagram as I and E. E stands for exon, and I stands for intron. And introns are actually trimmed out from the fully functional mRNA that you see down here at the bottom that are going to be exported through a nuclear pore into the cytoplasm. In the cytoplasm, we encounter what? Ribosomes that can translate, okay? But we have to have fully processed messenger RNAs to do that. And so they've got like little scissors here to show you the processing of the RNA, where the introns are cut out and the exons are spliced together. Now, those are usually not protein enzymes that do that cutting and splicing. It's usually RNA enzymes. Oftentimes they self-splice and self-ligate into fully functional mRNAs. Now, you might say, if you've got a eukaryotic DNA here, and you've got the messenger RNA that's transcribed that parts of it aren't used, why do eukaryotes put those pieces in? You know, does that make sense? Doesn't make sense at all, and didn't make sense to scientists for a long time. They figured there must be a reason why all our genes are littered with these introns. And they started figuring out what some of those things are, okay? So some of the functions of introns are as counters. Right? So if you've compartmentalized your nucleus, right, you got this true membrane around it, and you're making an mRNA that's going to come out here in the cytoplasm and be translated into protein, when it gets out here, how do you know how much you've got? If you're in this compartment, how do you know how much is out here in this compartment? Well, if you leave a little piece of it behind, you can count those. Right? You can get a concentration of those that will turn off the transcription of that gene. Okay? Another way, or another reason to have these introns is that, okay, let's just say that when I'm making this messenger RNA, it's to utilize a specific metabolic pathway, or it's to do something. Like maybe it's for the catabolism of a sugar. Maybe I want to utilize one sugar, right? And I don't want pathways to be on for the utilization of other sugars. If you have this intron left behind, guess what? It can act as a regulator, turning off those competing pathways. So it can function to bind to and inhibit, right, competing pathways, competing metabolic pathways. So those are a couple of reasons. Scientists have found additional reasons, but we don't need to go into all of them. But those are a couple of reasons you might have intron DNA. Now, if we wanted to clone a eukaryotic gene into a prokaryote, which you guys know we've done it with a lot of human growth hormones, insulin, blood clotting factors, and things like that you can get medically. They're produced in bacteria in mass quantities and provided usually at, fairly minimal cost to us as compared with the past where they were isolated from either usually animal sources, and they weren't as biocompatible with us either. When you clone a eukaryotic gene, right, into a prokaryote, and it gets transcribed into messenger RNA, you can imagine if all these introns are there, it's going to get pretty screwed up. Okay? So what we have to do, actually, if we're going to clone a gene, is get fully functional messenger RNA, reverse transcribe that into DNA, and then clone it. Okay? So it requires some thinking about introns and exons when you do gene cloning. All right, so any questions about transcription? No? All right, translation. So When we translate, remember, we're going from RNA to protein. Now, I should mention here that there are a large number of RNAs in the cell that are never translated. Okay? So non-translated RNAs. Can you think of any non-translated RNAs? Anybody? Anybody? Bueller? No. What are some other RNAs you know of? This is what? Messenger RNA? mRNA? tRNA isn't translated. Ribosomal RNA isn't translated. There are microRNAs, which you guys will hear about, that are involved in RNA interference. That you'll hear about when you get to molecular biology that aren't translated. So there are a lot of RNAs that don't go through this process. But there's a specific kind that do, and they're called messenger RNAs. Messenger RNAs have on them what we call a leader sequence, okay, that I mentioned just a minute ago, which means translation doesn't start at the very beginning of messenger RNA. It actually starts somewhere down the road, okay, down that sequence. There is a place where ribosomes made of ribosomal RNA and protein, and these are very, very large complexes, about 50 proteins and several RNAs make up these complexes. And the ribosome will actually come together. These subunits, the small ribosomal subunit and large ribosomal subunit that you see here, come together on that messenger RNA, and they assemble there on that leader sequence. And part of that leader sequence is called the Shine-Dalgarno sequence. Okay? And so that's where the ribosome will bind, and then it'll start sliding down the transcript until it gets to the start site of translation. Okay, and you guys know what the start site is? It's a specific codon, all right, which is a 3-nucleotide sequence, and it encodes for methionine. So if you guys see here, remember, AUG, right? So it slides down until it gets to the methionine. Now, is methionine always at the start of a protein? Yes, it is. In a eukaryote and in archaea, it's always at the start. It may not be the start of the final functional protein, right? There may be some processing that goes on of that protein, some trimming, so that it's not the start, but it always starts off that way. It's going to start off the protein sequence before it gets trimmed up into the fully functional protein. In bacteria, is that always the start? It's going to be the same start codon, different amino acid, okay? But it's not too different. It's just a modified, okay? So in bacteria, instead of a methionine, okay, you're going to get something called F-met, or informal methionine. Okay, so a modified methionine is incorporated for prokaryotes. And that's one way that they differ from eukarya. And if you guys remember the branch of that tree that I showed you earlier, we had a branch, right? We had bacteria, over here, we had eukaryotes over here, and then there was a branch that came off the eukaryotes that were the archaea. Right? So I said there were some molecular processes that suggest eukaryotes and archaea are more closely related than eukaryotes and archaea are to bacteria, even though archaea look structurally a lot like bacteria. They're single unicelled organisms, right? But this is one of those molecular processes that make them look distinct. All right, so the ribosome assembles, and deep down in the pocket of the 50S ribosome, we get the transpeptidase reaction, which is where the peptide is formed, right? Peptide bonds are formed between amino acids. That's where they're covalently joined together, deep down in this binding pocket of the 50S ribosome. Now, what we're going to get is a growing amino acid chain that comes out this tunnel in the 50S ribosome that gives us the new nascent polypeptide chain. And it's at that point in time that this peptide is going to be folded. Now, it's only going to exit that ribosome once in a lifetime, right, for that peptide. And so when it comes out of that tunnel, it's when its shape is dictated. It's dictated by the primary sequence of amino acids, which give rise to basically the chemical environment it's going to see as it exits that peptide. Right? And so any hydrophobic interactions, any covalent bonds between sulfurs, any ionic interactions need to happen there. So this is where folding is going to occur right as it exits that tunnel. Now, what if there's misfolding? Okay, or what if it gets misfolded later? Have you guys heard about proteins that fix that? Yeah, so there's some chaperoning proteins that can fix that. They're called the GRO-ES in prokaryotes, proteins that fix messed up proteins, but we really, really rely on them coming out of that ribosome and being fully functional when they exit through that tunnel. Okay, so let's look at translation. Okay, so as the ribosome finds that first methionine and moves along, it encounters a bunch of nucleotide sequences, right? And it wasn't clear initially when scientists looked at nucleotides how they were going to correspond to a protein sequence, to an amino acid sequence, because there's no chemical complementarity, right? And DNA sequences, nucleotide sequences, tend to be a lot longer than protein sequences. And so there were some experiments that went on to figure out how this occurred, but there were some really interesting ones that allow us to determine that there's a genetic code where 3 nucleotides correspond to the production of, or the incorporation of a single amino acid in a protein sequence. And these were done by a couple of guys called Nierenberg and Matai. And they actually won the Nobel Prize, and they elucidated this genetic code over a period of about 5 years. And let me show you what they did, because it's pretty darn cool. I actually love the story of it. So Nierenberg and Matai, at the time, they could isolate nucleotides, right, individual nucleotides, and put them in a test tube or an Eppendorf tube in the lab. And so to that tube, they might add a nucleotide like dideoxy adenosine, right? So you have maybe A's, or you could put T's. I'm not T's because they're not part of RNA. U's, right, or G's or C's, right? And they had an enzyme that they had isolated, which could, in a sequence-independent manner, link these nucleotides together, okay? So what you could do is add this enzyme, and you would get a string of A's, okay? So in this test tube, you'd have a whole bunch of A's, or you could have a whole bunch of T's, right? And so then what they did was they isolated this extract, and they threw in a cell extract that had ribosomes in it, okay? And then what they added to the mixture was a whole bunch of amino acids that were radio-labeled. Okay, so they were tagged. So what do you think happened? Well, the ribosomes would recognize these new RNA sequences that had been produced in the test tube. There were a lot of them there. The ribosomes would translate them using the amino acids that they found in the test tube, right? And then the scientist would isolate any polypeptides that came out of this, okay, that were radio-labeled, and then they would analyze them to see what they were made of. And so if we go back to that genetic code, right, when you add all adenines, right, to a test tube, you get a sequence of AAAAA. What they ended up were producing peptides that were only made of lysine. Right? When they added all C's, they got peptides that were only made of proline. When they added all U's, peptides that were only made of phenylalanine. And when they added all G's, peptides that were only made of glycine. All right? And so they thought, oh, we're on to something, right? And so then what they did was they started making combinations thereof of the nucleotides and seeing what polypeptide chains, what amino acids are in the polypeptide chains that lead us to getting these things. Okay? And so they, over a course of about 5 years and a lot of experiments, worked out this genetic code. They found that methionine was always used as a start, right? And they found a whole bunch of codons, right, when they had them, didn't produce any peptides. And so they were stop codons, but they called them nonsense codons because they didn't make any sense at the time. So we had sense codons, all the ones that produced a polypeptide, and then we had nonsense codons, which were the stop codons. They also found the degeneracy of the code, or the wobble, where the wobble says that at the third position, sometimes it doesn't matter what that third nucleotide is, right? So, for instance, if we look in this situation, UC, UCA, or G will all produce serine, okay? So it doesn't matter what the nucleotide is at the third position. That's not true of all of these, but it is true for a lot of them. Now, do all organisms use the same genetic code? First question, do we all use the same amino acids? For the most part, right? We use the basic 20. Some organisms also use selenocysteine and pyrrolysine are found in the archaea. So those two amino acids are actually incorporated at stop codons that are near a sequence-specific DNA region that allows their incorporation into a stop codon. But we're not really going to go into how those two are incorporated. But yeah, we do use basically the same genetic code, except for certain organelles like mitochondria, right? Use a different set of stop codons. But there is some degeneracy in the code. So, and there's also what we call codon bias. So say, for example, and I'm going to use this as an example, we look at this leucine here, CU and then whatever. All right, let's just say that if you're human, and I don't know if this is true or not, I'm making it up, but it's just for an example. If you were, for example, human, humans might use this codon 95% of the time, and those other codons the rest of the 5% of the time, okay? So what it amounts to is having more tRNAs that are for this particular amino acid than the other codon sequences, okay? So let's just say you're a bacteria. You might use CUG 95% of the time, right? And these others 5% of the time, okay? So we may use the same sequence, but we may not use them in the same proportion, right? And sometimes that has to do with how our genes are regulated too. So if you don't want high-level expression of a particular protein, you know what you can do to its gene sequence? Put in one of the codons that aren't used as frequently, and that slows down the process. So you can control the rate of translation by choosing specific codons because of the codon usage of an organism. Do you guys understand that? Ask me if you don't. No, I don't get that.
Why does it slow down?
So let's just say 95% of the time in humans, we use this codon, right? And so we have a lot of tRNAs that recognize this codon that can incorporate leucine. And we decide that we need to downregulate that sequence, right? Maybe that protein doesn't need to be made at that high levels. We want to sort of tweak its expression. What can happen is we'll end up maybe with a mutant, right, that incorporates instead of CUU, CUA or CUC or CUG, which are only found in 5% of the tRNAs that could be recognized. All right, so now what happens is when the ribosome comes across that particular codon, it has to sit there and wait longer for the diffusion of the right tRNA than it had when 95% of them, when it had the sequence where 95% of them would recognize it. So it slows down translation. So you can actually control the rate of translation by the codons that are chosen to be used in the genes or evolved to be used in the genes. Does that make sense? There are other ways to control the rate of translation. That's just one. The rest of them you'll probably talk about in molecular, especially if you take it from me. So you guys have heard, some of you guys have heard this already. All right. So next, what connects the codon sequence with the amino acid sequence? This was proposed by Crick, Watson and Crick. He happened to be right. He called it his adapter hypothesis. Okay, and it turns out that there's an RNA molecule that can be loaded with an amino acid, okay, by something called a tRNA synthase. And so what a tRNA synthase does, it takes a specific tRNA that has a certain nucleotide sequence, and pairs it with a particular amino acid, okay? And so here we see what tRNAs look like. They have, if you draw them out with their base pairing, they look like a clover leaf with a whole bunch of stem loops. They usually have one arm that has something called the anticodon, which can base pair with the codon specifically, right? But at the other end, they have an acceptor arm, which is an amino acid binding site. So if you look at a space-filling model of a tRNA, it actually has an L shape. One little piece of it serves as the anticodon arm, the part that can recognize the codon, and the other arm down here, which will be in that pocket region of the 50S ribosome, right, is going to contain the amino acid that's specific for that anticodon sequence. All right, so there's a tRNA that's going to match, tRNA synthase that matches these up, the amino acid with the codon. Okay? Now, so this picture is just to remind you guys that we have several different types of RNAs out there. We have the message, our translated RNAs. We have RNAs that serve as adapters, the tRNAs that connect codons and anticodons, and our ribosomal RNAs that form part of the ribosome that actually catalyze the peptidyltransferase reaction deep down in the pocket of the ribosome. Because remember, the ribosome, I mentioned this before, is a ribozyme. And translation occurs, this is a snapshot in time, just like RNA polymerase, we had a snapshot in time. We call this a polyribosome, where we've got the 5' end of messenger RNA, the 3' end down here, and you see multiple ribosomes moving along this transcript once it's made, making new protein. Okay, and here's a scanning EM of the same thing. I love the scanning EM because you can actually see both subunits of the ribosomes in some of these, and you can tell which is the 5' end and the 3' end. See, the protein's nice and short down at this end, but gets very, very long down on these ribosomes, right? So you can actually see, you know, new protein being made and amino acids and those little bead, you know, beads on a string look. All right, so what happens first? Well, the ribosomes assemble on the Shine-Delgarno sequence. They travel down to that first methionine. That first methionine is here, AUG. And what happens is we wait for the diffusion of a tRNA that carries the appropriate amino acid. And that's the rate-limiting step of the reaction. The ribosome just sitting around waiting for this diffusion. Okay, and probably why the ribozyme or ribosome is a ribozyme and not a protein enzyme. Protein enzymes tend to catalyze faster than RNA enzymes. But speed is not the rate of the enzyme is not the rate-limiting step of this reaction. It's the diffusion of tRNAs to that particular ribosome. Okay, so what will happen is that tRNA diffuses. It will base pair the anticodon sequence with the codon sequence, and then we wait for the diffusion of the second amino acid here, which happens to be attached to, or the tRNA that's attached to asparagine. Okay, now they show the peptide bond formation out here. You guys know it's deep down inside the pocket, but for picture purposes, they drew it outside of the enzyme itself, but it's deep down in that catalytic pocket. And then what happens is the ribosome is actually putting a kink in the mRNA right there, right between those two codons. And when it slides, right, it'll slide 3 amino acids and kink the RNA again, right? And that's how the codons are separated from each other. It's by the ribosome moving and putting a kink into the mRNA. And so it sort of ratchets along, okay, at a certain, distance. And as it does that, the sliding, right, the methionine is trimmed from the tRNA, and the tRNA goes on to be used again. We don't waste it, right? It goes out there in the cytoplasm and encounters another tRNA synthase, which can reload it, okay? It's kind of like a phone card. You can reload it with a few dimes. You know, you can reload it with a few an amino acid, so it can be reused again. And then we wait for that tRNA carrying our third amino acid, our next codon. And so this progresses along all the way down the transcript. We get the synthesis of the polypeptide chain and its folding. And then how do we know that we're at the end? Okay, another question. Scientists looked for years to see if they could find a tRNA that recognized stop codons. Because that's what would make sense, right? When you come across a stop codon, you have a tRNA that recognizes it that just doesn't have an amino acid attached to it. Okay, that's what scientists thought they were going to find, but no, that's not what they found. So it took them a little bit longer because that's what they were searching for. They found these things called elongation factors, or EFs. Okay. I'm sorry, it's not elongation factors, it's RFs, release factors. So there are 3 in bacteria to recognize the 3 different stop codons. Okay, so there are actually proteins that bind to those stop codons that prevent the movement of the translational apparatus. So you guys got any questions so far? We're going to stop a little early tonight and start lab a little early because I think this lab actually takes some time, so I want to get started a little bit earlier. So I'm going to give you guys like a 10-minute break or so, and then we'll meet in lab so that we can make sure we get done in time tonight. We've got a bunch of stains to do. Okay?